All right, everyone. My name is Joseph Zinsky. I am a postdoctoral researcher here at the University of Pennsylvania. And today I'll be talking to you about CRISPR-Cas9 and the interesting sub-story that's buried within CRISPR, which is really why we work on basic science. And I don't mean basic science by really simple science. I mean basic in terms of there are really two types. Basic means fundamental to how we understand science as a whole. And then there's translational, which is trying to cure cancer, trying to cure disease, trying to find a better way to modify the genome, which is what they were initially trying to do before CRISPR came along. And a lot of times we look over fundamental research because we think, it's not going to directly get us what we want. But in many cases, it can lead to some of the greatest discoveries because of the interconnectedness of all of biological science. So, before we understand, in order to understand and appreciate what an accomplishment CRISPR was, you have to really appreciate how hard it was to make mutations back before we had direct genome editing with CRISPR. So if you wanted to make a knockout, say, the first thing you want to do in genetics, you find a gene, you want to study it, you want to knock it out, right? You want to see what it does when it's gone. So if you wanted to do that, you couldn't just go in and edit the genome. You would have to essentially perform an entire forward genetic screen. So forward genetic screens, you have your Drosophila, you feed them some sort of mutagen, its gametes accumulate a bunch of random mutations, cross it out, have flies that each have some assortment of mutations, look at the F1s, see if there's any dominant mutations, cross them back in, look at the F2s, see if there's any recessive mutations, do this thousands upon thousands of times, and hope that one of those will end up being the gene that you initially wanted to study. And you wouldn't even know which one was the gene you wanted to study. You'd have to go back and individually genotype each one of those flies, map the mutation, and find where it was. So most fly nomenclature is all done in terms of what the fly looked like, because at the time nobody knew what gene they were getting. They just knew that they had a fly that had white eyes, or they had a fly that was deformed, and they named it as such. Later on, they found where the genes were by mapping and things of that nature, but it takes some time. And so if you wanted to work on a model organism that didn't have a forward genetic screen done with for it to do evolutionary development or something like that, you were just kind of out of luck. So a long time, biologists have wanted to be able to go into the genome and just make these edits, right? Just as a side note, I mentioned that EMS is the way that you can introduce mutations into the Drosophila gametes, but there were also screens done using P element insertions. I wanted to touch on them just because we're going to be talking about transposable elements again later in the talk, and you should probably all know what they are anyway. So transposable elements look like this. You have tandem inverted repeats on either end of a length of DNA. In the center, sometimes it will encode a transposase, sometimes it won't, but that transposase, wherever it ends up coming from, will bind to these tandem inverted repeats, bring them together, and pop the DNA out, and then paste it somewhere else. There are some transposases which will copy and paste as opposed to cut and paste, but essentially P elements are genetic elements that are able to just jump around the genome. And you can imagine that's very useful if you want to disrupt genes instead of having a mutagen that causes a bunch of DNA damage. You can instead hop a P element around at random and see if that integrates into a gene, and then look to see if that's disrupted the gene. So these are two ways of doing forward genetic screens, but they're all long, they're all arduous, and so the field really wanted a way to say, here's a gene I want to look at. Let's knock it out. Let's get a mutation in there. So that is the story of why we want genome editing, and we're first going to go into how we were trying to push forward with genome editing, and then this second strand of research, which is the fundamental research on bacterial immunity, and then those two things converge and ultimately give us CRISPR gene editing. So the story starts, one of the first really effective ways that we had to edit genomes where and when we wanted to edit them was using homologous recombination. This was first done in yeast and cell lines. It was later pretty quickly adapted to mice, but it took a long time to get it into Drosophila, and it really wasn't widely used or very effective, which we'll talk about why in a second. So what does using homologous recombination to edit genes look like? So you have your gene of interest. You want to insert something in there. You would design a plasmid that looks something like this, right? You'd have length of DNA that's homologous to one side of where you wanted to insert, a length of DNA that's homologous to another side of where you wanted to insert. These homology arms will allow it to pop in here eventually. You'd include your desired insertion as well as some sort of marker. In this case, it's going to be Uro3. Uro3 is the gene that yeast needs to make its own uracil. We'll talk about why that's important in a second. And then you would want to get it into your cell. Say we're using yeast. You want to get it into your yeast. The way to get plasmids into cell lines, one of the ways, is using electroporation. It's a way of disrupting the plasma membrane of a cell, exploiting the fact that there's a voltage differential across cell membranes. And when you introduce a current across there, you cause the cell to have its membrane rupture, trying to equalize the charge. And while that's happening, the plasmid that you introduce can go into the cell, and then it heals up afterwards. So, okay, some percentage of time, you're going to get integration via homologous recombination, which we've covered early in the course. And that's great because this Uro3 gene, which the yeast needs to make its own uracil, you were growing your media, you were probably growing your yeast in a media with uracil in it. Now you take the uracil out. Only the yeast that have had successful integration will continue to grow, and you can use those because you have your knockout. So why was this, what problems were there here? I told you that it took a long time for it to be implemented in other model organisms. I told you it didn't really get a lot of widespread use outside of cell lines. And that's because successful homologous recombination mediated integration is extremely infrequent, something to the tune of 0.1%. You have to put it into thousands of cells and hope that one gets a successful integration, which you can totally do if you're using a cell line because you have thousands of cells. But now, say you wanted to put it into an animal. Well, if you wanted to put it into a Drosophila, even if you were to express it in a Drosophila oocyte, you can't screen through thousands of embryos. That's just impractical. So it was a wonder that we got it to work in animals in any real way at all. So yeah, what if we want to edit an animal's genome? Well, mice had an interesting workaround because you can culture mouse embryonic stem cells outside of the mice. And if you have a cell line, you can do the same trick we just did with the yeast, except with different markers. Transform your plasmid into the embryonic stem cells and end up with a population of mouse embryonic stem cells that are in a plate outside of the mouse. And one of the cool things about mice is that you can actually culture mouse blastocysts outside of the mouse as well, and then inject these cells into the blastocysts. And this population of cells that exists inside of the blastocyst are pluripotent. They're embryonic stem cells, and they have the potential to make any tissue in the organism. And so when you inject these cells in, some population will take hold and go on to make some part of the mouse. It's a really cool process. I mean, you can see it here. This is a mouse blastocyst being held in place by suction, and now they're injecting the cells into the blastocell cavity. So once this happens, how do we know if our injected cells successfully populated the embryo? Well, they use a pretty cool trick for this, and that's that they'll usually use a white mouse blastocyst and a black mouse embryonic stem cell. And so if any parts of the mouse that came from the embryonic stem cells that were from the black mouse will show up with black fur, and any parts that were from the host white mouse that are wild type will show up in white. And you end up with what we call a chimera. This mouse is part wild type and part whatever embryonic stem cell edited thing that we introduced. And so we can't use the chimeras, but the chimera has some black fur, some white fur, but it's also going to have some of its gametes be either one genotype or the other, either from the black mouse or the white mouse. And so when you cross it back to a white mouse, now any gametes that came from the black mouse will be totally black. Any gametes that came from the white mouse will be totally white. You can take the black mice and you have your population of edited mice. So mice were able to, mouse genetics really thrived on the fact that you could do this for a long time, and it gave it a big leg up on other model organisms for doing reverse genetics. And I said forward genetics when I was doing screens. Forward genetics means that you start with the phenotype and you find the gene. Reverse genetics means that you start with the gene so that you can study the phenotype. And so mice were able to do reverse genetics. All right, so this was great. We had homologous recombination mediated repair, but it had its flaws, and everybody else wanted a piece of the action. So one of the next major breakthroughs in this this journey was the discovery of non-homologous end joining. So non-homologous end joining is the alternative repair mechanism to homologous recombination, and people quickly found that they could utilize this to make a variety of deletions in genomic DNA. So the advantage here is that when you introduce a cut, now non-homologous end joining comes to repair things, and it doesn't do a good job. And so these modifications that are introduced by non-homologous end joining are much more frequent than by homologous recombination. Maybe 10%, 20% of the cut sites will have a small deletion or a small insertion. And if you put in some donor DNA just at random, some amount of that will end up getting inserted. And then they thought, well, what if we combine this with homologous recombination, introduce a cut, instead of having it repair itself through non-homologous end joining, which it'll do if it doesn't have another copy, right? We could introduce our own DNA that has homology arms. Now the cell wants to repair itself. It sees that there's donor DNA with homology arms, and it uses that to integrate. So they were able to use this to make large or small insertions at a much higher rate than homologous recombination alone, because the cell is actively looking for a way to repair the damage that has been introduced here. So this is great. If we could do this, then a success rate of 10 to 50% is more than enough to put it into the oocytes of all model organisms and perform reverse genetics on flies or whatever you really wanted to do. But, how do we make this DNA damage happen where and when we want it to happen? How do we introduce the cuts where we want them? So the first answer to this question came with the discovery of zinc finger proteins. So zinc finger proteins were domains of transcription factors that had these zinc fingers, and they were used about a decade later as a way of targeting nucleases to create DNA breaks. So zinc fingers are the DNA binding domains of a pretty expansive family of transcription factors. Each of these zinc finger protein domains recognizes a specific 3 base pair sequence. The transcription factor will have many of these lined up. It will allow it to recognize many different sequences and localize, in the case of the transcription factor, an activating domain to the promoter of a gene or whatever it's trying to do. But scientists quickly realized that they could string together themselves a bunch of these zinc finger domains, and make something that will target a number of different combinations of base pairs. It can't do all of them. There are, I'm not sure how many 3 base pair combinations you can get, but probably 90% we have a zinc finger domain for. So this is great. You could now target these strings of zinc finger proteins to different parts of the genome. And then if you attach something like a FOC1 nuclease to it, and put half of the FOC1 nucleus on one zinc finger and half the FOC1 nucleus on another zinc finger and have them sit down on adjacent regions of the genome, when they come together, they'll make the cut, and that will cleave the DNA. And now you can successfully introduce DNA breaks where you want to introduce them, and then do that whole trick with homologous recombination if you want to do an insertion. So the major shortcoming here was that it was pretty difficult and time-consuming to make these. You'd have to clone an entire protein every time you wanted to make a new zinc finger. This took time, and it really didn't catch on. It still took, 3 months if your lab had all of the zinc fingers already had the DNA for it and worked on the first try and everything was going well. And if you didn't know, it could take upwards to like 6 months to a year. So that was just too long for it to really catch on. So the next big breakthrough came in 2009, 2010, where talins were used to target these nucleases to where they wanted them to go. So talins are a really cool example of fundamental basic science bleeding over into this translational science before CRISPR did it. So tail effectors are from Xanthomyces aureus. They're a plant pathogen, and they inject these tail effectors into the host that they are infecting. These go into the host nucleus and activate some host genes that make the infection easier for the bacteria. And the cool thing about these tail effectors is that, unlike zinc fingers, which recognize triplets of DNA, these have domains that recognize a single base pair. So, GACT. So there's only 4 flavors of these, and you can string as many as you want together to make a talin that can recognize any sequence of base pairs. Again, you string on some FOC1 endonucleases, and now you can cut any base that you want in the entire genome. So this was a little bit easier to do than with zinc fingers, because rather than having to have a whole library of 50 to 60 of these zinc fingers, now you only need 4. There's 4 domains that go into making a talin, and that simplifies the process of making it. Still, you're making a protein every single time, and that means that there is a chance that it'll fail, and it is still quite time consuming. So just as talins were looking like they were maybe going to catch on, CRISPR burst onto the scene. Before we get to that, a small aside. So we're talking about making small insertions or deletions, and you have the problem of figuring out, well, how are we going to detect these small insertions or deletions in the genomes of the cell lines or the animals that we're working with. So usually the talins will have a success rate, or CRISPR will have a success rate of about 10%. If you wanted to detect that, there's really 2 ways to go about it. If it's a big insertion, you could PCR across the insertion site and then run it on a gel and observe differences in sizes, because the bigger stuff will run slower and the smaller stuff will run faster. And you will see the size difference of the ones that successfully received an insertion from your break and then homologous recombination. But what if it's smaller? What if it is only a few base pairs? You didn't put in any DNA to do homologous recombination with. Now it's going to be a deletion or of an insertion of a couple of base pairs caused by non-homologous end joining, and PCRing across the site will not give you any spread when you run it on a gel. So what you do is you still PCR amplify it, but then you denature and anneal by heating to break everything apart, and then cooling to allow the strands to re-anneal with each other. Some of the strands will re-anneal wild type to wild type, some will re-anneal mutation to mutation, and importantly, some will re-anneal with a wild type adjacent to one of the mutations. And there's a specific endonuclease called T7 endonuclease 1, which will cut any mismatched base pairs. And then we run this on a gel. For the wild type, you'll see 1 band. If you have successfully made some cuts, you will see multiple bands, as it will have been, some percentage will be digested by this endonuclease 1. So both of these are your really simple checks as to whether your TALEN or zinc finger or CRISPR is working properly. So let's change gears from this area of research on genome editing to one on bacterial immunity that was happening in parallel. So the first event didn't really have anything to do with bacterial immunity at all, and that was just when genomes were being sequenced, people were noticing that there were lots of these mysterious repeat sequences, clustered, regularly interspaced, short palindromic repeats, as they were dubbed. So these CRISPR repeats were discovered in the late 80s, but it wasn't until the mid-2000s until their function became more clear. So CRISPR locus, when discovered, looked something like this. You had a bunch of conserved repeats, and in between those repeats, there were these unique spacer sequences. They were identified by the conserved repeats, but it was the unique spacer sequences that ultimately gave away what the function might be. And that's because it was shown in the mid-2000s that these unique spacer sequences actually match the sequence of bacteriophages, specifically double-stranded DNA bacteriophages. This clued everyone in that somehow the bacteria was voluntarily integrating bacterial DNA into its own genome in these regular arrays. So the search for what proteins could be performing this function began, and the ones that were first looked at were the ones that were closest by. These were termed the Cas proteins, and it was soon found that Cas1 and Cas2 are the two genes that are required to get this bacterial phage DNA and integrate it into the unique spacer sequence regions. So it does so by like this. So Cas1 and Cas2 are produced. They form a pretty cool looking complex. Two Cas1 dimers and one Cas2 dimer form this tetrameric complex. And this complex, as depicted by its crystal structure here, is shown holding a 23 base pair region that it would have excised from a bacteriophage. And this is what it looks like when it's traveling around and grabbing things and performing its function. So Cas1 and Cas2 will go find phage DNA, cut out a chunk, and bring it back and integrate it into the host genome in this CRISPR array. Simple as that. But really, you should be asking yourself, well, how do Cas1 and Cas2 differentiate from phage DNA and bacterial DNA? Because if these two proteins are nucleases, which they are nucleases that recognize double-stranded DNA, cut them up and bring things back to the CRISPR array, well, there's got to be some rhyme or reason, or else it would be cutting up the bacteria's DNA and bringing that to the CRISPR region, which would kill the bacteria. So the answer comes in these double-stranded breaks. So when a double-stranded break happens in bacteria, RecBCD are recruited to repair the site. RecBCD perform repairs by chewing the sequence back towards one of these Chi sites. Chi sites are areas in the bacterial genome that are designated for homologous recombination, and they can be used to repair the sequence that has been lost. So RecBCD, as it is chewing back towards one of the Chi sites, releases a lot of single-stranded products. And these single-stranded products are what Cas1 and Cas2 recognize and bring back to the CRISPR array. So the next question then is, well, okay, so this implies that there must be more double-stranded breaks in the phage DNA than there is in the host genome, or else it would still be chewing up the host genome more than the phage DNA. This happens and is true for a number of reasons. The first of which is the most basic defense mechanism that bacteria have against phage invasion, and that is the production of restriction enzymes. You've probably talked about them in the class already. They're XO1, BAM1, they have names like this. They probably were introduced as something that chews up double-stranded DNA. Set sequence, CCTTT, CT something is going to be recognized by a particular restriction enzyme, and then it will cut somewhere along there. Well, the vast majority of these restriction enzymes were found in bacteria in the wild, and that's where we got them. We now use them for molecular biology. When you want to make a cut, you find a restriction enzyme. But initially, this was a bacterial defense against phage DNA. So essentially, it works like this. The bacteria would carry a restriction enzyme that recognizes a particular sequence. It would also carry a methyltransferase that would methylate the bacteria's sequence that matches what the restriction enzyme would cut. The restriction enzyme would not be able to cut the bacteria's DNA because the methylation would stop it, but it could cut the phage DNA because the phages do not methylate their DNA. So this would introduce a lot more breaks into the phage DNA and less into the bacterial genome, which would again lead to more breaks and more integration into the CRISPR locus by Cas1 and Cas2. So we talked about restriction enzymes. The next thing is stalled replication forks. So replication is an inherently messy process, which tends to lead to double-stranded breaks. When these occur, RecBCD has to be called in, again, causing there to be fragments that Cas1 and Cas2 can take back. And finally, bacteria has a circular chromosome where the phage will have a linear chromosome, and these ends of the linear chromosome of the phages will always be recognized as double-stranded breaks and can be chewed back to a chi site. So I mentioned that DNA replication is something that causes double-stranded breaks. What I didn't say there is that there will be more replication forks in the bacteriophage because bacteriophages have multiple origins of replication, whereas a host bacteria will have one origin and therefore two replication forks. The phage will have many different origins and therefore many replication forks and chances for things to go wrong. The other interesting feature of phages is that they have very few chi sites. So whenever RecBCD is recruited to a phage, it has to chew things back very, very far in order to induce homologous recombination and try to repair. And this will produce a lot more fragments and give Cas1 and Cas2 a lot more chances to work. So phage DNA has more origins, it's vulnerable to restriction enzymes, it's linear, it has less chi sites, and therefore it is has more double-stranded breaks, which recruits Cas1 and Cas2 to it. That's not to say Cas1 and Cas2 is perfect. There will be times when Cas1 and Cas2 accidentally takes a piece of the host genome and brings it back to that site. And when that happens, it's very bad. But the cell at that point would just die. And there are lots and lots of prokaryotic organisms, and that's something that happens from time to time. Things kill themselves accidentally. But when you have lots and lots of things, then it's not so bad. So one kind of interesting point is that phages are in somewhat of an arms race. I mean, viruses and hosts are always in arms races to try to counteract each other's defense mechanisms. And some phages do try to methylate their own DNA to stop bacteria from cutting them up with restriction enzymes. But phages tend to have very small genomes, and they therefore will not be able to carry a lot of different methyltransferases to methylate their own genome, and therefore they have trouble methylating their own DNA sufficiently to defend against all the possible attacks that they could face. But how this is done is still poorly understood, but it is kind of an interesting thing that phages do try to methylate their own DNA. Okay, so why is it so bad when a piece of the host genome gets integrated into one of these CRISPR arrays? Well, that's because many of the Cas proteins encode putative helicases and endonucleases, which Cas1 and Cas2 are both endonucleases. Cas1 has some helicase properties where it can unwind the DNA. Helicases are proteins that can unwind DNA. Endonucleases are proteins that can cut the DNA. And ultimately, the CRISPR system functions as an adaptive immunity for bacteria against phages. It works like this. Cas1 and Cas2 go and identify phage DNA, bring it back to the CRISPR array. Then the other Cas proteins are produced, and they come and get these unique spacer sequences, which have been put in there by Cas1 and Cas2. They combine with a specific protein. They combine with a specific protein. Sometimes it's multiple proteins. In this case, we'll be talking about Cas9 since that's the most common one. Cas9 will come and pick up one of these CRRNAs and go back to the phage and chop up the DNA with its own endonuclease activity. So all in all, the Cas1, Cas2 identifies bacteriophage DNA, integrates it into the host genome, and the host genome then expresses these DNA sequences as small RNAs that are integrated into a complex that can go back and cut up the phage DNA whenever they see that particular phage again. So all in all, it's a really cool defense mechanism against phages. But you can also see why if the Cas1, Cas2 were to integrate part of the host genome into itself, it would constantly be chopping up its own genome and lead to the death of the host. So it's very important for Cas1 and Cas2 to identify the phages correctly. So what advantages do CRISPR have over restriction endonucleases? They're both defense mechanisms against phages, but they kind of shine in different scenarios. So when there's low parasite diversity, this is when CRISPR is at its finest. The bacteria will encounter a single type of phage. Part of that phage will then get integrated into the CRISPR locus, and now the CRISPR will chop up anytime it will chop up the DNA anytime it sees that phage again. Whereas restriction enzymes may hit that phage and may not. You could get unlucky and you're just not carrying the restriction enzyme that happens to chop up that phage's DNA. Plus, you're carrying a whole bunch of different restriction enzymes, which is a bunch of energy that the cell has to produce that has to waste producing methyltransferases and the restriction enzymes themselves. Whereas the CRISPR, you have one set of proteins that's being produced and it will recognize that phage. There's always a cost to producing more things. CRISPR is also better when bacteria try to pass beneficial plasmids to each other. At times, there will be plasmids that confer immunity against antibiotics or against phages. Bacteria will pass that from bacteria to bacteria with cool little tubes that we're probably not covering in the course. But the CRISPR, these plasmids tend to not be methylated and therefore might get chopped up by restriction enzymes, whereas the CRISPR will not recognize and chop up that particular plasmid. But there's one scenario where restriction enzymes tend to be better, and that's when you're seeing a very diverse array of different viruses. So if you're always seeing a new virus, it doesn't really help you to integrate the viruses into your genome and try to recognize it because you'll never see it again. In that case, it's really better just to have a bunch of restriction endonucleases and hope like hell that the virus that you get infected with happens to be one that you have a restriction endonuclease against, and if not, you'll end up dying if you was a bacteria. Yeah, so this is the sort of the conundrum and the reason that bacteria probably have developed both restriction endonucleases and a CRISPR system to work together. Okay, so to recap, we've learned that CRISPR is an extremely effective bacterial immune system, and its response can really be broken up into three parts. There's the CRISPR adaptation step, where Cas1 and Cas2 excise some phage DNA and insert it into the CRISPR array. Then there is the expression and processing step, which we didn't talk about really at first, and we'll talk about that in a sec. And then finally, there's an interference step where the phage is chopped up by this effector complex. That's a combination of these RNAs that are expressed in the CRISPR locus and the Cas proteins that are expressed in the Cas operon. So let's talk a bit about these precursor cRNAs. So the CRISPR RNA, the CRISPR locus is transcribed in its entirety, and this long RNA is broken up and turned into these little unique RNAs with a stem loop at the end. These stem loops are important for the structure of the ultimate cRNA effector complex when it combines with the other Cas proteins. So we have the CRISPR locus, it's expressed in its entirety, that's chopped up. Each one of those finds its own set of Cas proteins, and they all go off to fight the little phages. But this CRISPR has been discovered in a wide range of different species of bacteria, and these different bacteria use different Cas proteins to perform these three main functions. So this is the current classification of the known CRISPR systems. And so let's break these down. So in our CRISPR adaptation step, Cas1 and Cas2 are really well conserved, probably the most conserved of any of the CRISPR elements. Sometimes you'll see a Cas4 protein, but you'll always see Cas1 and Cas2. And Cas1 is really the most important of these. It performs the helicase activity of unwinding the phage DNA. It performs the endonuclease activity of cutting the phage's DNA. The Cas2 dimer is really acting as a spacer in determining how big this length of DNA that's going to be excised is. So Cas1 is probably the most important of this first step. In the second step, Cas6 or RNase 3 is able to cleave these precursor cRNAs into fully formed mature cRNAs with the stem loop. And then finally, Cas9 is the protein that you've probably heard before because it's called CRISPR Cas9 colloquially layman. It's called Cas9 in layman terms. I can't pronounce things apparently. But in other bacterial systems that have been discovered, you could have a range of different proteins coming together to form a kind of a Voltron to mimic Cas9's function. So in a type 1 system, you'll have Cas7, Cas5, SS, Cas8, and Cas3 all coming together with the cRNA to perform the interference step, where in the type 2 system, Cas9 can perform all of those together. So that's one of the reasons, well, that's the main reason that Cas9 is used for gene editing, and why Ishmael will be talking about it when he goes over how we're utilizing it in the field to edit genomes. But it's worth taking a look at this diverse range of other proteins and thinking about how this system could evolve. So how did the CRISPR system evolve? So I told you that mobile genetic elements would come back up in the talk, and now is that time. So mobile genetic elements constitute about 30% of the prokaryotic genome and about 50% of the human genome. Before, we mentioned P elements, which are DNA transposons that are able to cut and paste or copy and paste themselves around a genome. But there's other types of transposable elements. There are retrotransposons, which instead of staying as DNA, they create an RNA, which they can make many copies of. And that RNA, by reverse transcriptase, makes a DNA, and then that integrates into the genome to paste itself. There's also plasmids. Bacteria can share plasmids. We mentioned mutualistic plasmids when we were talking about CRISPR. Passing this DNA from bacteria to bacteria also constitutes being a mobile genetic element. And finally, and most importantly for the evolution of CRISPR, is viruses like phages introducing transposons that can hop around. So a big breakthrough in understanding where CRISPR came from was realizing that in some bacteria, Cas1, instead of residing with all the other CRISPR proteins near the CRISPR locus, is actually far away in the genome. And these faraway Cas1s were flanked by repeating elements that resembled transposons. They named them Casposons because they contained a Cas1 protein. These Casposons contained a Cas1 protein, DNA pol B, which is a polymerase that viruses often use, and they were flanked by repeats that resembled the inverted repeats seen in DNA transposons. Some of them even contained capsid proteins, which a virus would include in one of its transposons because it would want to make more capsid proteins to replicate once it integrates. So this gave a lot of clues that Casposons initially came from a viral integration of a viral phage element. So when they analyzed the DNA of these Casposons, they found that they were in fact related to the Cas1 that we see in the CRISPR arrays. So this is a phylogenetic tree. I'm not sure if you've seen them before in the course, so briefly, they estimate evolutionary relationships. They're often constructed based on DNA sequence similarity, and the one that we're looking at is based on DNA sequence similarity. And essentially, things on the left are ancestors, and things on the right are things that exist in present day. So everything here that has a name is a current bacteria that has a Cas boson, or Cas1. And everything on the left would be proposed ancestral splits that diverged into different species, or different Cas bosons. So the length of the lines implies differences in sequence. So Cas1F and Cas1E have a shorter line that connects them than Cas1F and Cas1B, and therefore they are more similar in DNA sequence. And if they're more similar, they're assumed to have a more recent common ancestor. So one note, this is not an actual proxy for time, though elapsed time can be estimated. It's more about sequence similarity. So we can assume that Cas1F and Cas1E diverged more recently than Cas1F and Cas1B because their sequences are of F and B are more disparate than F and E. But how long ago those splits occurred is unknown. It can really only be estimated. You can estimate it based on the known rate of mutation in DNA or something of that nature, but it's not something we can tell precisely from just the DNA sequence alone. That said, what all of this shows is that CRISPR-Cas1, these Cas bosons, diverged a long time ago from the CRISPR-Cas1s that are near the CRISPR locus. So something about landing near that CRISPR locus was a fortuitous event that allowed Cas1 to start to take on a new function separate from these Cas bosons, which really don't do much of anything. So what might have happened here? Well, we have this mobile genetic element of Cas1 and Cas2 being introduced by a prophage. And in some case, they think that this Cas1, Cas2 mobile genetic element happened to integrate next to an innate immune response system that already existed in bacteria, containing the other Cas proteins that was essentially acting like RNAi or a restriction enzyme, taking a sequence and then cutting up whenever they see that sequence. So the only difference here would be there would be no Cas1 and Cas2, so you wouldn't be putting new sequences into the Cas boson. You would just be having some set sequence that the bacteria always knew to attack. But once Cas1 and Cas2 integrated, the ability of that mobile genetic element to jump was lost, along with the viral capsid proteins. And Cas1 and Cas2 took on a new function, integrating phage DNA into the CRISPR array. So what's yet to be discovered? There's quite a few things. This is an incomplete picture and a best guess as to how CRISPR may have evolved. But the big things are that Cas bosons with Cas1 and Cas4 have been discovered, but none with Cas2. So we're not really sure where Cas2 came from, if it integrated later or if it was initially part of the Cas bosons and was just lost in some of the other ones. And finally, I described Cas1 and Cas2 sitting down next to some innate immune response, but no one has discovered this innate immune response without Cas1 and Cas2 present. So those things still have to be figured out. And you might think, well, how can we not possibly have discovered, this innate immune response? We've sequenced so much things, we know so much about science. Well, about 98% of the bacterial genomes in the world have yet to be sequenced because there's so many species of bacteria. So most likely, the bacteria that have these innate immune responses without Cas1 and Cas2 are out there, and we just haven't looked at them yet. So work on bacteria. It's very fun. You learn lots of stuff. Okay, so after Cas1 and Cas2 integrate next to this immune response, and the initial CRISPR system starts to function, it's thought that CRISPR over time got more and more simple. So the class 1, type 1, type 3, type 4 were the ancestors, and over time they evolved into the type 2 system, which is the most simple system that we've discovered so far. mostly because it has this one giant protein Cas9, which performs all of the steps of interference, and that makes it ideal for genome editing. Let's talk a little more about this wonder protein Cas9. So Cas9 is the protein that binds to the mature cRNA and uses that to recognize phage DNA to go off and perform its interference functions. But prior to doing all of this, Cas9 also plays a role in processing the precursor cRNA. So the precursor cRNA is a read across the entire CRISPR array. So it's going to include the unique spacer sequences as well as the conserved repeats. Cas9 uses a track RNA to bind to the conserved repeats. So the track RNA is produced in the CRISPR locus. It performs a number of stem loops, and those stem loops help to bind to the Cas9 and form a proper structure. Those stem loops, those track RNAs also have regions that hybridize with the conserved repeats in the CRISPR array. So that helps to guide Cas9 to the pre-cRNA that is produced from the CRISPR array. Once it's there, RNase 3 comes and cuts, separating each of the Cas9s from each other, and each Cas9 then has a unique spacer sequence buried into it, or embedded into it, that it uses to go off and perform interference functions. So yeah, I mentioned RNase 3 or Cas6 depending on the system, but for the system we're talking about, RNase 3 cleaves these pre-RNAs and makes mature RNAs that are already embedded within the Cas9. So the track RNA forms these stem loops. It hybridizes with the fragment of the conserved sequences in the pre-RNA. Inside the Cas9, you have a 20 base pair unique sequence, as well as a fragment of the conserved part of the CRISPR array that's hybridizing with the track RNA. All right, so I mentioned that now Cas9 is toting around this 20 base pair complementary fragment, which it will use to find DNA and cut it. So how does it go about doing this? Well, rather than searching along all the different DNA, there's one very important feature that the Cas9 is always searching for. And that feature is this thing called the protospacer adjacent motif, or PAM. PAM is a NGG, which means any base pair followed by a GG, that must always follow the 20 base pair target. So Cas9 moves along DNA searching for PAM sequences, and whenever it encounters a PAM sequence, it checks to see if the 20 base pair complementary fragment it's carrying hybridizes with those 20 base pairs that precede the PAM sequence. And so ultimately, this allows Cas9 to more quickly find potential sites where it should be testing whether or not its complementary fragment binds. And this is actually extremely important for preventing the destruction of the CRISPR locus, which we'll talk about in a second. So once it finds its PAM sequence and the 20 base pairs have successfully hybridized, Cas9 has two endonucleases, the RUVC and the HNH, which cleave 3 base pairs upstream of the PAM sequence. So if this is your PAM sequence, you're going to be cleaving that 3 base pairs upstream. So I mentioned before that the PAM sequence is really important for making sure that CRISPR doesn't destroy its own locus. And so that's because each of these unique spacer sequences that are 20 base pairs are contained in the CRISPR locus. So if all Cas9 needed to do was recognize 20 base pairs, it would always recognize the CRISPR locus as being foreign DNA and chop up the CRISPR locus, killing the bacteria every single time. What the PAM locus does is allow for the CRISPR locus to not be destroyed. So inside of these conserved repeats, there are no PAM sites. So even though the 20 base pairs would hybridize to these unique spacer sequences, There is no PAM sequence in the conserved repeat, and therefore Cas9 does not chop up its own locus. Between 2005 and 2012, CRISPR had essentially gone from a bacterial immune system to becoming a fast, easy method to edit genes. So let's recap how genome editing has evolved so far. We started out with homologous recombination that had the shortcoming of being very low frequency, so having limited use. Through research on DNA repair, we ended up realizing that utilizing homologous recombination or non-homologous end joining combined with a DNA break could increase the frequency. And so then the problem became, well, how do we make targeted DNA breaks where we want to make them? Through another unrelated area of research into transcription factors, there was the discovery of zinc fingers. Zinc fingers offered the ability to target DNA breaks where we wanted to make them. But creating zinc finger transcription factors from the 64 possible zinc fingers was really time consuming, and so it was difficult for labs to utilize. Then from another disparate area of research, plant bacterial research, there was the advent of talins. Talins, rather than having 64 possible zinc fingers, only had 4 tail effector domains that could be put together to create a fully formed talin, so it was easier to make than a zinc finger. But it was still time consuming because you had to make a new protein every single time you wanted to target a new region of your DNA. And so now along comes CRISPR from yet another disparate area of research in bacterial immunity. And CRISPR's big advances was that now all you need is this single Cas9 protein to make all the cuts. And what you have to remake each time is a cRNA, so a new RNA for targeting the Cas9. And making RNA is much, much easier than making protein. A matter of days as compared to a matter of weeks. Hell, you can make an RNA in an afternoon. It's very, very fast process, a couple of hours. As CRISPR started to be utilized more and more as a genome editing tool, they were looking for ways to simplify the system even further. And so one of the big ones was that you had to make your Cas9 protein, you had to make a cRNA, and you also had to make this track RNA. And so you're having to make 2 different RNAs. And so they found that you could actually link these 2 RNAs together so that instead of making 2 RNAs, you could just make 1 single RNA. And so here's a plot showing a portion of DNA in lane 1 where you introduce Cas9 but no guide RNAs and there's no cuts that are made. You can introduce a cRNA and a track RNA and it is cut. Or you can introduce a single chimera where the 2 RNAs are fused together by a GAA linker, and that performs the same function of making the cuts in the necessary place. So a single RNA could cut anywhere in the DNA. So that's 1 protein, 1 RNA, make targeted cuts anywhere you want. So just as a reference, this is the part of the conserved CRISPR locus. This is the 20 base pair unique thing that you're trying to bind to. This is the linker region. And this is stem loop 1. And you'll notice the track RNA usually has 2 more stem loops. But they found that only stem loop 1 was actually necessary. So in reality, you didn't even need stem loops 2 and 3. And now when you create a CRISPR RNA, you only use stem loop 1. And this is but stem loop 1 is in fact necessary. And this is a chimera that is lacking stem loop 1, and that one does not make the cut. So you at the very least need stem loop 1. Indispensable for CRISPR function. So CRISPR has been the area of heavy research, and therefore there's been a lot of crystallography done on it. And so they were able to crystallize the protein structure as well as the protein while it had the guide RNA and the target RNA inside of it. And when they did so, they were able to kind of see why only stem loop 1 was necessary and not stem loops 2 and 3. So you're probably not going to get tested on this, but it's just really cool to see. So this is stem loop 1. This is the linker loop. And this is the actual 20 base pair guide. And when you have the host DNA here in black and gray, you can see the black part hybridizing with the 20 base pair guide, the other part being melted away and put to another part of the protein. And this is what Cas9 looks like when it's surrounding this RNA DNA complex. So you can see now why stem loop 1 is the most necessary. Stem loops 2 and 3 interact relatively minimally with the host, the DNA that's being melted. So they're not required for Cas9 to perform its activity, but they do increase the efficacy if they're there. To sum things up, I think this is a really beautiful story illustrating how we go from basic science to breakthroughs that are going to be applicable to editing the genomes in research and possibly one day helping to cure a disease by using a viral delivery of the CRISPR system into humans. It's going to win a Nobel Prize. It's one of the most influential things to happen in science in a long time and has changed almost every field. But it comes from bacterial research and transcription factor research and research on DNA repair and research on plant bacteria, all these things where they were not trying to revolutionize genome engineering, but without a proper understanding, we would never end up with CRISPR gene editing, right? And aside from it being many different areas of research coming together, it's also many different regions, labs in all different parts of the world contributing to this, all across America, Europe, and China. So next week, Ish is going to talk a bit more about the applications, about how you can use Cas9 to activate and repress genes or target proteins to places, edit base pairs, make things fluoresce, edit RNAs. There's all kinds of things you can do with the Cas protein system, and it truly is revolutionizing many areas of science. So these are the references where I pulled a lot of the figures from. Some of them are the original papers where CRISPR were discovered. A lot of these other ones are reviews. I would highly recommend, if you want more information, this CRISPR-Cas9 Adapting to Change in Science. It's a great overview of everything that's happening with the system. The Kunin and Makarova paper on origins and evolution of the CRISPR system is also particularly good if you're interested in that and more about that Evo-Devo. My name is Joseph Zinsky. Again, you can email me with any questions at joe.zinsky@gmail.com, and I'll be happy to get back to you.